in case I don't see this particular peak, so then this tells me that this amino acid, 161 glutamine, was exposed to the solvent. And it does not, suppose I have a molten globule structure of a protein, so there's going to be some amino acids which are exposed outside, and there's some which are going to be inside. The ones which are outside, they can undergo an exchange with the solvent, where I've used D2O as my solvent. So these hydrogens which are coupled to the nitrogens, they will get exchanged. So then they will not appear in the HSQC. So first I will record a Barnett chain of 15 and HSQC, label all my peaks, and then I'm going to replace my solvent with D2O, then record an HSQC again, and in that I will see that some peaks will be missing. So the peaks which are missing, they will tell you the amino acids which are exposed to the solvent, because of which they have undergone an exchange and they do not come with the HSQC. So basically the H in HSQC, it gives us information about the chemical environment of each nuclei and the correlation between them. So it basically tells us the correlation between them, meaning that it tells me the Raman frequency of the hydrogen and the nitrogen, and it tells me the chemical environment of each of these amino acids. Okay, so now, so from the 2D NMR, what is the information we've got? We've got the information if we have got that this nitrogen is coupled to this nitrogen is coupled to this hydrogen, then I go back to the protein data bank and I can find out which of these which this NH corresponds to which amino acid. So then now, suppose I want to find out the sequence of a protein. So now if I want to find out the sequence of a protein, it's not sufficient if I just have the NH. I should know the C alpha to which this nitrogen is connected. So this means now I need the Raman frequency of the hydrogen, I need the Raman frequency of the nitrogen, and I also need Raman frequency of the carbon. So now I need to measure in 3 dimensions, which cannot be done in a 2D NMR. So that's when you go to a 3D NMR. So this will tell you, since it gives you a correlation between the Raman frequency of hydrogen, nitrogen, and carbon, you can get the entire sequence of the proteins. So this is basically a 3D NMR. So now I'll just quickly give a quantum mechanical description of NMR. So we initially spoke about these two states, the plus half and the minus half. So in quantum mechanics, this state is known as the alpha state, and this state is known as the beta state. So this is denoted, the alpha state which is denoted by plus half is a spin-up state, and the beta state which is denoted by the minus half is a spin-down state. This is the Hamiltonian for a single spin, which is equal to the Raman frequency times Iz. Iz here is the angular momentum component about the Z. This is the Hamiltonian for two spins, which is equal to the Raman frequency times the angular momentum of first spin into Raman frequency of second spin into the angular momentum of the second spin, and this component is a coupling constant between both of them. So this is the matrix notation for the Hamiltonian of a single spin. So these are my states, the alpha. So this is how matrix notation is generally written in quantum mechanics. So then, so now when I solve this, when I do quantum mechanical calculations and I solve this, I get the energy. I get the energy level of each of these states. So this is of the alpha state, and this is of the beta state. And then if you see the difference between these two states, like this comes out to be equal to gamma HB, which is what we had got even from the initial slide. When I showed you delta E, I got the same answer. So basically it's that, the classical picture and the quantum mechanical picture matches. Yeah, so far we've discussed for a single spin. I've just told you for a single spin, if it's spin half, it's going to split up into two. But then now my sample contains a lot of spins. There are a lot of spins in my sample. So it's not, it's no more possible to explain with this spin diagram. This we've just done it for a single spin half. So that's when you introduce a quantity known as the density operator. So NMR is a bulk phenomena, meaning it involves millions and millions of spins. So to represent the state of the system, we need an ensemble average. And this ensemble averaging is contained within the density operator. So the density operator for an ensemble of single spin half nuclei can be expressed as a linear combination of these matrices, which is Ix, Iy, Iz, and E. Ix is the angular momentum operator bar the X, Iy is the angular momentum operator bar the Y, Iz is the angular momentum operator bar the Z, and E is the unity units. And from quantum mechanical calculations, you can get the value of Iz, Ixn, and Iy. So the density operator, I have mentioned it is represented by rho, the symbol rho. So how we have a time-dependent Schrodinger equation which tells you the evolution of a state. This is the density operator. Similarly, there is an equation known as the Liouville-von-Neumann equation, which tells you how this density operator evolves. So this density operator has the information about all the spins. It's like an ensemble averaging of all these billions and trillions of spins which is contained in my sample. And this equation will tell me like how the TDSE, the time dependent Schrodinger equation, tells me how the wave function evolves. This tells me how this density operator will evolve. So again, if you do some quantum mechanical calculations, you will get that at equilibrium, the density matrix will contain only the IZ term. That is, at equilibrium, you will have magnetization only along the Z. And this is what we had got even from our classical picture, where we have 60 spins here and 40 spins here. The net magnetization was along Z. So again, if you do some quantum mechanical calculations, you get that the density matrix, which is the ensemble average, will contain, you get the magnetization to be only around Z. Yeah, so this is a density operator for a two-spin system. So these are the product operators for spin 1 and spin 2. When you multiply each of these operators with these quantities, like the I2x, I1x with I2x, you get I1x, I2x, I1x, I2y, I1x, I2z, and I1x, E2. So you get 16 of them. Even in E2 will just give you a unity matrix, and that is neglected. So you get these 15 operators. So this I1z is described as the Z magnetization on spin 1. The I1x and I1y are the in-phase X and Y magnetizations on spin 1. I2z is the Z magnetization on spin 2. I2x, I2y is the in-phase X magnetization and Y magnetization on spin 2. It's the same as this, but here instead of it being spin 1, it is spin 2. The next is you get some anti-phase terms. I1x, I2z and I1y, I2z are the anti-phase X and Y magnetizations on spin 1. Similarly, when this coefficient 1 is replaced by 2, you will get the anti-phase X and Y magnetizations on spin 2, and you get some multiple covalence terms and the non-equilibrium population. So using these product operators, you can now explain the HSQC pulse sequence. So basically, we know from quantum mechanics and from this classical picture that the initial magnetization is about the Z-axis. So initially, I start off with the Z-axis, and I apply this 90-degree pulse. So this 90-degree pulse, it brings my Z magnetization along the Y. So now I have in-phase Y magnetization over here. Now the part A is known as a spin echo. So what this basically does is that the in-phase Y magnetization, which is there over here, it will convert it here to the anti-phase X magnetization. So this at the end of this, I have anti-phase X magnetization on spin 1, and now when I apply an 90-degree pulse to both the spins, this anti-phase magnetization gets converted from one spin to another spin, from the first spin to the second spin. So what HSQC is a 2D experiment. So we are trying to get information about both the nuclei. Like, if I have a hydrogen and a carbon, I'm trying to get the Raman frequencies of both the carbon and the nitrogen. But then when I record my data, this is the FID, I'm recording it only along the along the hydrogen. Like say my spin 1 is hydrogen and my spin I is hydrogen and my spin S is nitrogen. So I'm going to be recording data only from the hydrogen. So what I do is that I try and acquire information about the hydrogen through this pulse sequence. Then in between I transfer magnetization from one of the spins to the other spins, and then I try and get information about that spin, and then I transfer this magnetization back to the first spin, and then I finally detect the signal. So what finally the signal will have is the information about the first spin and the second. So yeah, that's all.
Questions? Okay, so now Shwetha will show us presentation, right? So she has a presentation of the Lindau meeting. So yes.
So this year on the first week of July this year, I attended the Lindau Nobel Laureate Meeting. So this was the 63rd Lindau Nobel Laureate Meeting, and it was dedicated to the field of chemistry. The major topics on focus were energy conservation and structural biology. So this meeting was attended this year by 35 Nobel laureates, among which 27 were from the field of chemistry, 6 of them were physicists, one was a Nobel Peace Prize winner, and the last one was from physiology and medicine. So even though it's dedicated to the field of chemistry, the the Lindau Council tries and makes it as interdisciplinary as possible. So Lindau is basically it's a small island which is present in the south of Germany, and it is bordered by Lake Constance. So it is the same lake which borders in Austria and Switzerland. So apparently you can get to Austria and Switzerland by boat in a couple of hours from Lindau. And it's a very important trading sector and a tourist attraction. So these pictures are the landmark of Lindau, and you must be wondering why I put the same picture 3 times. It's just that this landmark, this is a lion and this is the lighthouse. So during each time of the day, this looks extremely beautiful and at the same time different from each other. So basically the selection procedure, there are 2 rounds of selection. So in about the second week of August, the DST sends out an invitation for applicants to apply. So anyone who is pursuing bachelor's, master's, PhD or postdoc is eligible to apply for this meeting. So all you need to do is you need to fill a form, and you need 2 recommendation letters, and you need to send this to DST. So you need to send it by October, by the second week of October. So then the DST, what it does is it scrutinizes all these forms, and they bring down they shortlist the number of candidates to 30 from how many applications they received, and these 30 candidates are then nominated by the DST to the Lindau Council. So that's the second round of selection. So the Lindau Council, in the council are some members who are organizing this, and 15 Nobel laureates. So again, they scrutinize these forms, and they bring down the number of candidates from 32 to 25. So the everyday schedule was like we had scientific breakfast. So the council had planned it in such a way that you spend your entire day with Nobel laureates. All your meals, your breakfast, lunch, dinners, their talks, everything is all of them are with the Nobel laureates. So we had something known as a scientific breakfast. So in a scientific breakfast, what happened was that a topic was taken up for discussion, and since the meeting was focused on energy conservation, most of the topics in the scientific breakfast were on energy conservation. So there are round tables which are put up, and on each table is a Nobel laureate. So you can go and you discuss with them about whatever issue they're talking about. There were talks on what chemists can do for conserving energy and such and so. So this happened for about 2 hours, and after 2 hours, each person from every table they summarize what was discussed. So and in between you're allowed to switch tables as well. If you think that you contributed to a table and you want to attend another discussion, so you can switch to another table where another Nobel laureate is sitting. So then there were lectures, discussion sessions, panel discussions, master classes, and formal dinners. So the opening summary was the best part of the meeting. So the opening summary was scheduled for 30th June and it was supposed to happen at 3 p.m. So we had to get ourselves registered by 10 p.m. and all of us did that. And the island is very small. It's just about 0.7 square kilometers by area. So you can just walk around for 2 hours and you've seen the entire place. So that's what we did. Once we got ourselves registered, we saw around the place. And then the opening summary was scheduled for 3 o'clock. So all of us decided that we have a quick lunch and go and get seats in the meeting hall. So I had my lunch, and I think about 2 o'clock I was there in the hall. So I decided to sit in the first few rows, and it was during that time that I saw that the 4th row was entirely empty. So I thought I'd go sit over there. So when I went over there, one of the people from the Lindau Council, the organizing committee, came and told me that you're not allowed to sit here. This row is reserved for the Nobel laureates. So that's when I came to know that they were going to sit in row 4. So I quickly got myself a place in row 3, so that I could have the Nobel laureates sit right behind me. So correctly when the clock struck 3, there's a song which is played and all the Nobel laureates enter. And this was indeed the best moment during the entire meeting. I mean, some of the Nobel laureates are really old. There was this physicist called Alex Muller. So he can't even walk by himself. So there was another Nobel laureate who was helping him walk. And there's this famous chemist who won the Nobel Prize for catalysis, Hertel. So he is extremely old. So he walks with a stick. So he was walking slowly, and there was another Nobel laureate helping him walk. So this happened right at 3 o'clock, and sitting right behind me was Priyanka Kovilka, who won the Nobel Prize last year, and this is Countess Bettina. So her family are the sponsors for this meeting. So this meeting has been happening from 1951, and initially it was only for the Germans. So the Nobel laureates who took part were Germans, the students who came were also Germans, and was during the course of time they realized that the Germans realized that it's important to find out how science is being done in other countries as well. So that's why they declared this meeting open to other countries. So it's her family which has been sponsoring this ever since. I think her grandfather started it, she is Astrid Grassman. So she is at the Stockholm University. So she was also in the organizing committee, and she's right now in the selection panel of the Nobel laureates. There are 5 people who formed the final selection panel, and she's in the selection panel for chemistry. So the lectures. So the first lecture was by Professor Priya Kovilka. He won the Nobel Prize last year for studying the structure and function of G protein coupled receptors. It's a very important transmembrane protein. So since he had won the Nobel Prize just last year, it was his first time as well in the meeting. So and this talk was supposed to be the first, was the opening talk of this meeting. So that was on the 1st of July, and it was supposed to be at 9 o'clock. So around 8:40 or so, I went to the hall, and I again got a seat for myself. And around 8:45, he goes up the stage and he starts pacing up and down. So all of us are wondering what's wrong with him. I mean, you don't imagine a Nobel laureate to be tensed when he's giving a talk. And then soon, in 5-7 minutes, you could realize that he was tensed. And when he came up, the first thing he said was that, I'm sure all of you have realized by now that I am tense, and he accepted that he was extremely tense since he was a new kid to this club of Nobel laureates. That's how he described himself. So, and undoubtedly he gave a brilliant lecture about his Nobel discovery. Then a talk by Richard Ernst. So Richard Ernst spoke about the importance of extracurricular activities in one's life. So he won the Nobel Prize in 1991 for NMR, for Nuclear Magnetic Resonance Spectroscopy, for the development of this technique. So in this talk, he gave us his own example. So what he said was the fact of using Fourier transformation. For NMR, he came to know this when he was attending a concert. So when he was listening to music, that's when he came to know that the instrument, which every instrument has its own frequency, and this, so whenever I hear music, it can be broken down to different sine and cosine waves. And another example, interesting example he gave of himself was during the 1970s or so, he and his wife, they happened to come to Asia. So they traveled, they went to India, China, and then after they went to Tibet. So when they were walking through Tibet, they saw that on the roadside there were a lot of these Tibetan paintings. So he and his wife really fell in love with these paintings, and they bought a huge lot of them, and they went back to Switzerland. So he's basically from Switzerland. And there, then in a book he read about the importance of these Tibetan paintings. Like the pigments which are used, they have some special quality. So he started. Since he had these paintings, what he did was he started doing experiments using Raman spectroscopy on these paintings. And ultimately, this led to him owning a Raman spectroscopy in his garage below his house. He has a Raman spectrometer, and so with this paintings, he takes infrared images of these paintings. What these infrared images are. So the color from the painting is removed. So if a person so you can just see the outline, and if something is written on the painting, you can see that. So apparently, according to Tibetan history, in art, only the master is allowed to do the painting. So the master will draw, and he will fill colors. He will write colors like la, ma, ka. So la means yellow, ma means red, and the students are allowed to fill colors. So they are not allowed to draw. So he could see these letters la, ma, ka written in those paintings. And that's when he went and read more about Tibetan philosophy, and now he's an expert in that. And this is Serge. He was one of the physicists who gave a brilliant talk on the quantum world. And this is Ada Yunath. Now she told us something very interesting. So she won the Nobel Prize for the crystallization of ribosomes. So when she took up this problem for research, all what she heard was that drop this problem. Ribosomes cannot be crystallized. A lot of people have been working on this for a very long time, and they've given their life on this. So you should just drop this problem. So the main point why they can't be crystallized is that they disintegrate. Before the crystal is formed, these ribosomes disintegrate. So she was considering to give up this, and that's when she came across an article. So this was an article on polar bears. So what that article read was that in these polar bears, the ribosomes, they do not disintegrate. So what happens is that these polar bears during the season of hibernation, they do not need any proteins. So all these ribosomes, they come together and they form a complex, and this prevents the ribosomes from disintegrating. And then once the hibernation is over and these polar bears come out, now if the ribosomes are disintegrated, then it's going to take a long time for the body to make ribosomes and then synthesize proteins. So then this is the way the polar bears are born, such a way that the ribosomes come together, form a complex, and they do not disintegrate. And once the polar bears come out of hibernation, this complex just breaks and the ribosomes start performing their functional synthesizing. So this gave her an idea of how she could use the fact that the ribosomes do not disintegrate. And she won the Nobel Prize in the year 2011 with Venki Ramakrishnan. I'm sure all of you must have heard of him. So they won the Nobel Prize for solving the structure of ribosomes using X-ray crystallography. This is Professor Aaron Chekanover. So he is a very inspiring personality, very charismatic. So I had lunch with him on 2 days, and during one of the conversations, I just happened to ask him that what he thinks India should do, in the field of science, so that we contribute as much as the best does. So he told me what he thinks. He told me it's his personal view. He thinks that this country, India, he thinks that it lacks role models. He said this country lacks role models. So I asked him what does he define as as someone, what you need to do to become a role model. So he tell me what people do in this country, not just him, scientists do, is that they have a lab and they train students to the best they can, and then they send these students to the best universities for their postdoc. And these best universities include the top 5: Stanford, MIT, Harvard, Cambridge, Oxford. So these students they do a long postdoc there for 5 years, 6 years, and then during the training period itself they are told that whatever you learn, how much ever you find science to be better in that country, you're supposed to come back to your country and do science there. So he won the Nobel Prize with his PhD guide. So his PhD guide did that to him, and now he's doing that to his students, and apparently students have also started doing that to their students. So it was very good talking to him. And Kroto gave a talk on presentation skills. So he is one of the best speakers I've heard. He is very, very good. So I had on a personal discussion with him. I asked him some questions on endometrial fullerenes. Fullerenes is a C60 molecule, the buckyball. So he won the Nobel Prize for discovering them. So I had some questions on endometrial fullerenes, and I went and asked him that. And then he told me that his postdoc is taking part in the meeting. He said he works on this field, so you can interact with him. So his postdoc's name was Paul, and we didn't know where Paul was, so both of us started looking for him, and we found Paul. So I started talking to Paul, and then I was asking Paul about Kroto. So Paul was saying that Paul is right now a postdoc, but he's been doing from the time of his bachelor, he's been there associated with Kroto's lab. So he was telling me that Kroto is an extremely busy person. I believe he's so busy that he doesn't have time to read papers which the student write, and so on and so forth. So what Paul has done is he's created an email ID which has the name of Kroto. So he sends his own, whenever he's finished the paper, he sends it for submission, and whatever reviews come, he makes changes and he resubmits it. So then he was telling me this, and he was pretty disappointed about it, that Kroto was so busy. So I just happened to ask him how research is going on and how he's doing. He said it's going decent. I've published 2 PhDs this year, 1 JAX is under review, and I have to go back and finish another JAX. So he's been doing all this independently by himself. So apart from this, there were panel discussions. So there was one panel discussion was on why communicate, and the importance of English in science was discussed. So all the panel are all Nobel laureates, and the audience is free to ask them any question. And there was something also known as a master class. So what exactly is a master class? Is that anyone who's selected to take part in the Lindau meeting can apply to a Nobel laureate. So every meeting there are few Nobel laureates who take classes. They teach their research field. So this time there was Guthrie. He won the Nobel Prize for protein NMR. So he was giving a master class. So you basically apply to them, and there's again a selection procedure. So there are 2 kinds of selection which are done. One for people who are selected to present their work to the Nobel laureate. So first for half an hour, 45 minutes, the Nobel laureate teaches you the basics of whatever he works on. And then for the next 45 minutes, 50 minutes, students who are selected present their work. And the next round of selection is where you get to audit. You just get to attend the class and the research. So I was selected to audit this particular master class which was taken by Guthrie. So during this, so after this master class, Guthrie actually saw the structure of the protein which I'm working on, the human prion protein. So he saw the first NMR structure came out by him. So I went and asked him some questions about this protein, and our discussion just shifted about how science is done in India. So then suddenly he came and told me that during this discussion that he thinks that Indians don't do enough science. He said science is not done properly in India. I mean, it's not done enough. So I asked him what makes him say that. So he told me that he said that he's been coming to India for a long time now. He's been a regular visitor at the IISER, NMR Research Center, and he feels that people don't work enough. According to him, they come to the lab in the late morning and they leave by late afternoon. And he said this is not enough. So I refused to agree with him. I told him that's not how things are done. So then he asked me, how old are you? So I told him I'm 20. So then he asked me, he said, so do you work on protein NMR? I told him I'm doing a summer at IISER. then he told me, have you recorded an NMR spectrum by yourself without the help of a PhD? And no undergraduate in India is allowed to do that. So I told him, no, I haven't done that. So he said, that's it. In my lab, apparently in his lab, he has two 16-year-olds who record NMR data by themselves, alone, without the help of a PhD. He just told me this and he walked off. And then a couple of days later, I met him again and he told me that the first thing I should do at IISER is that record data by myself, by myself. So when I came back to IISER, I told this to my guide. So my guide, Professor Atra, he knows Nuclear very well. I mean, they know each other very well. So he was going to work in protein NMR and I told him that this is what Nuclear has told me. He's told me to tell you that I have to record data by myself. So I told him it's up to you to teach me, get machine time, whatever book, and I have to learn how to run experiments. So luckily, Professor Atra, he agreed, and the data which I worked on during my summer project, I recorded all those data by myself, and I sent back a mail to Curie saying that I recorded my first NMR data. So yeah, on the last day from Lindau, we went to an island called Manau. So this Manau island is about 2.5 hours by boat from Lindau. So this was the last day, and this is where the closing ceremony of the Lindau meeting takes place. So this is the hotel where the Nobel laureates live. So from the Lindau island, we pick up these Nobel laureates, and we went to this island. So in this island, there was a panel discussion on peace. So this is James Bota. He is a Nobel Peace Prize winner who I can't feeling this evening, and there was a panel discussion on world peace. And this is a picture of me with Rudolf Marcus during the cruise. So I basically made a scrapbook, and the scrapbook had pictures of all these Nobel laureates, and I had planned to take their autographs. So the picture which I had of Rudolf Marcus was, I think at least when he was 20 or 30 years younger. So what he was telling me during this picture, the photo was taken, was that he has a lot more hair in the picture over here in the book than what he has over here. So in the Manau Island, there was something very interesting which I came to know. So they have a museum there. It's a part of the Nobel Museum, and it's a travel museum. So there's this artist by the name Berger. He's an artist and photographer. So what he did was, during one of the Lindau meetings, he got a message sent to all the Nobel laureates that there's something very important going to happen at 6 p.m. in this hall in Lindau. So all of you must come. So all the Nobel laureates, they obediently, they listened to him and they assembled in the hall. And what he did was he shoved all the doors of the hall and he gave them chart paper and crayon, and he said that you have to represent your work. And you're allowed to leave this hall only if you do this. So these are some of the sketches of some of the Nobel laureates. So this is of Ernst, who worked on 2D NMR. You can see the T1, T2. So again, he won the Nobel Prize for the development of the methodology of NMR spectroscopy. This is of Krotos. Like I told you, he discovered the C60, so green. And this is of Hartmut Michael, and he works on membrane proteins. So what he's drawn here is that these are lipid membranes.